consultant to Anchor Diagnostics. His work has significantly contributed to the fields of viral pathogenesis, molecular diagnostics, regulatory science, and international scientific collaboration. This evening, Dr. Alam will share his thoughts on the topic, Advancing the WHO R&D Blueprint for Viral Pathogens. Are we prepared for the next pandemic with precision diagnostics, next generation vaccines, and rapid therapeutics? His presentation will explore the evolving landscape of pandemic preparedness, the importance of precision diagnostics, and the growing significance of collaborative efforts between Germany and India in strengthening global health securities. Ladies and gentlemen, please join me in extending a warm welcome to Dr. MOHAMMAD INTAKHAB.
Very much for being here. I'm very pleased that you all made my expectations. Actually, I was not planned to deliver such kind of presentations here because it was planned in a very short time. And I realized that there is an importance in India that we have to connect science from Germany and we have to import the technology in India because we always dial up that we can manufacture, we have a manufacturing capacity, but in reality we are still have a lot of gaps. That's why to fill the gaps we really need to import the technology from our neighboring and partnership countries. That's why I organize these things that you all have the visibility, how we can achieve this milestone so that we can improve the healthcare ecosystem, because COVID-19 took millions of lives all over the world. Even doctors, pathologists, policymakers, even ministers, they did not save their lives during the COVID-19. The reason was that we were not prepared. We had no actual diagnostic that really needs to screen the patient, and also the vaccines or therapeutics we have to deliver to the patient, not only into the cosmopolitan cities but also into the remote areas. So that's why I really thought that we should organize such kind of things. But if you find any kind of lack in laxity in the organization, I really apologize that we did not make at that level or excellency, but we will try to follow. So I just want to say that here is my title, Advancing the WHO Blueprint to Viral Pathogens. Are we prepared for the next pandemic with precision diagnostics, next generation vaccines, and rapid therapeutics? As you know that I am Alam, and I share my residence in both the cities. One is Cologne, where I am permanently based, and it's more than 22 years I am living in this city. So I am also a German acquired citizen right now. So definitely I don't know how to say whether India is my first home or Germany is my first home. So definitely I would like to be loyal that I live in Germany and Germany is my first home and India is my second home. So and also I want to say that Hamburg city, this is the second largest city after Berlin. Berlin is a capital city and Hamburg is the second largest, and this is the gateway to the world because the ports and all the hub of the innovations are there. So this is the second largest city. So now I just want to say about myself that I did my PhD in medical virology at the Institute of Medical Virology from Eastern Germany. I did my 5 years postdoctoral position at my first postdoctoral was Institute of Medical Virology, Zurich, Switzerland, and other institutions in Europe, for example in Germany and Switzerland. So these are the two countries where I was most educated and trained. And I also did 2 years assistant professorship of virology at the medical school in Turkey. I was teaching medical student for medical virology. 2 years I worked as a CEO, Chief Scientific Officer of IVD, in vitro diagnostics in Germany. Since 8 years now, I am completing approximately 8 years in the in vitro diagnostic IVDs and medical devices such as CT scan, MRI, and also I am very expert in the regulatory affairs because India and Germany needs to be compliant in the regulatory matters. That's why I am also expertise in this area. Currently, I am also senior advisor for clinical trials professionals in Germany, both for preclinical and clinical trials. Whatever the basic science is going on at the academic institutions level, so that is called the preclinical. And when we translate the preclinical research into the clinical trials, that is human trials. And without human trials, we cannot understand the value of the preclinical. So that is a therapeutic, vaccines, and diagnostics. I also want to say that I am associate editor of the Journal of Cellular and Molecular Medicine. This is the European journal, and the impact factor is 5.5 or a little bit higher. This is a very prestigious journal. I am associate editor. I am also the director of Diagnomed GmbH in Germany and also in Delhi and Noida. So I founded this company in 2019 in order to connect science from European Union to India. So this was my mission to form this company. And now this is a real time that we have to start the ultra-fast PCR technology. Definitely my academic curriculum is fragmented, but it is extremely essential to contribute in many important sectors of the healthcare and research. You see here that India and Europe has a very strong dialogue to unite and tie for that's why innovator and technology innovator like Thomas Gravy and also Dr. RUCKES, TOBIAS, he is from Diagnostics from Hamburg. They are here. They want to fill the gaps. So that's why their technology is like they want to fill the critical gaps in the rapid PCR for fast patient health outcomes. Because PCR always takes, it is a complicated process. It requires manpower. It takes sampling and amplification and analysis. So altogether, I have been also doing a lot of PCR during my academic education, and it was not less than 4 to 5 hours or even 1 day. Now I understand that in India, I think most of the diagnostic laboratory is offering the PCR result in more than 3 hours or 4 hours. Even it takes overnight, and they deliver the result in 24 hours. So this is the situation we don't have to deal with. If pandemic hits, how you can wait for 12 hours? This is a very, very long time. That's why you have to screen the patient immediately, that somebody is infected or they are not infected. So 30-minute PCR is a technology that is a innovative technology which Thomas Gravy has invented. He is the innovator and innovator of the technology. What is existing, and he renovated the technology. So that's why you see here that our European perspective is here, and our Prime Minister Narendra Modi, they are tying up very strongly. Here is also our current Chancellor of Germany and all delegates from Delhi and also from Germany. They are also tying. Our Chief Minister is from Delhi. She is also very much supportive to establish the collaboration between European Union and India. So here you can see that India, EU has recently signed the agreement as a strategic breakthrough in India, global trade agreement. So free trade agreement, that means probably we don't need to pay the taxes to the both bilateral countries. Maybe the European products comes to India, might be the government does not want a lot of taxes, but they want to ease the process of importation and application into the healthcare system. The same way, India is also prepared with a lot of products that needs to be exported in European Union, so that the establishment of the collaboration will be really fruitful. So this is so now I want to say that rapid and ultra-fast 30-minute PCR innovative technology can significantly benefit to the patient diagnostic. First of all, I just want to say that immunocompromised patient, transplant, respiratory and autoimmune patients, and a lot of diseases are important for this kind of technology to apply for. As I said that I am also scientist. Actually, I have escalated and fragmented my positions in academic research, because without evidence, I don't believe something can be, convincing. So that's why I am a scientist, and I am an associate editor. Can you see that my name is here reflected? So I am an associate editor, and the editor-in-chief of this journal is from Belgium, Cancer Center, and also from the Cambridge. So this Stefan is the editor-in-chief of the scientific journal, and together with him we are accepting a lot of meritorious translational research from the academics and also the medical research. For example, All India Institute of Medical Sciences, NIA, National Institute of Immunology, ICGB, also Mumbai or anywhere the hospital is also being involved into the preclinical and clinical trials. So that's why I always believe in science. First I believe in science and then I try to integrate that science into the application of the human for the benefit. Now I want to say that what is a WHO blueprint pathogen? That is a priority pathogen. For example, COVID-19 is also under the blueprint pathogen. Crimean Congo hemorrhagic fever, Ebola virus, Lassa virus, Middle East respiratory virus, Nipah virus, Hanipa virus, Rift Valley fever, Zika virus, and this is a disease which is emerging every day. Every month it can come anytime. So that's why these are the red flags that indicate that this can cause a pandemic. Not COVID-19 alone, but this can come tomorrow. You never know that this disease can come tomorrow, and we can also have the same situations like respiratory, the COVID-19. So that's why we have to always work with the respiratory and other emerging viruses. WHO always update all the blueprint RNA pathogens list time to time, because one time they don't update, but time to time they update that which virus is a pandemic potential. Recent evidence shows that fungi can be more dangerous than viruses or bacteria, especially for ICU patients. You see here that they are a very elderly patient, they are also risk for the invasive fungal infection. So that's why I just want to say in this slide that blueprint viral pathogens means the pathogen. They have no capacity to be diagnosed. There is no therapeutics. There is no vaccine for such kind of because we don't have a therapeutics and vaccines or diagnostics. That's why we have to work on this front. I also want to say that there is some history of the viruses when they came. So I don't want to engage you in this understanding, but I want to say that after millions of years, now we are in a viral species. There are a lot of viruses in the environment that we have to cope up. You can see here that these are all global examples of emerging and re-emerging infectious diseases. These are club and half of the pathogens. You can see that how they are emerging, what time they are emerging, So these are also the reefs of the pathogens, the viral pathogens where they live, they remain, the viruses are in oceans, they are also in soil, they are in the fresh water, they are in us. So these are all the niche, you can understand the viruses are everywhere. Now I just want to say that these are the international agencies for the public health and regulation and future pandemic preparedness. So as you have heard about the CDC, Center for Disease Control and Prevention, there is a WHO, World Health Organization in Switzerland, EMA, that is the European Medical Agency in Netherlands, ECDC, that is the European Center for Disease Control, it is in Sweden, EU, CDC, African Union Center for Disease Control and Prevention, FDA, Food and Drug Administration, United States, BFOG, that is located in Bonn in Germany. Here is the Paul Ehrlich Institute in Germany, and here is PRECT, Preparedness and Resilience for Emerging Threats, initiated by WHO. So there is a collaboration of global experts, stakeholders, all the WHO members. So these international organizations are working all the time to develop the policy, how we can tackle the next pandemic. These are all international organizations. There's also 100 days. There is a 100 days project that is a priority research and development for vaccines, diagnostic and therapeutics in 100 days. For example, today there is a pandemic. We don't have drugs, diagnostic or vaccine. So Government of India, Government International Organization body, they want us and scientists that we have to develop the vaccines, diagnostic and therapeutic within 100 days. That is not possible. Not at all, because any kind of a drug or diagnostic or vaccines needs at least 10 to 12 hours as a roadmap to develop and to bring into the applications. So that's why this is a 100 days R&D project is not feasible or possible, but scientist has to work. Now I just want to say that Why we have to work for blueprint for viral pathogens? Because we have to first identify the pathogens. We have to do the research. Why such kind of viruses or bacteria or fungus is in our society? And we have to respond to dangerous viral pathogens that can cause epidemics or pandemics. The WHO R&D blueprint is a framework that accelerates research, development, and deployment of diagnostic vaccines and therapies for high-threat viruses, e.g., Ebola, Nipah, MERS, SARS, coronavirus. So these are all the pandemic potential viruses. Okay, now you can see here that zoonotic infections, the infections that is transmitted from animals to human. For example, rabies. Rabies is also one of the viral infections. I want to see the zoonotic infection. What is the zoonotic infection? Until now, we are not sure that the COVID-19 came from laboratory, or it came from the animal, or it was used as a recombinant virus for the bioterrorism. We never know. Even WHO cannot confirm today that COVID-19 was from the laboratory, or it was from the animal, or it was designed for viral spillover. So nobody knows. So that's why the zoonotic infection is the infection that comes from animal. So that's why we have to be isolated from the animal, habitat. Animals should be at a particular place, and our residents should be always at a different location. If we live together with the animal, there is a chance of the viruses that they can mutate and they can bind to the receptor and they can start infecting the human. So this can be possible, that zoonotic infection can come anytime. So that's why this is a very big project. I don't want to spend so much time, but our guests are also having a lot of time for explaining these things. So you can say that human can pass more pathogens into the animal than the animal pass to us. For example, the dog. The dog is passing a lot of viruses to us than what we passes to the dog. We never know, But that is true, and you can see that this is from England. In the Liverpool University, the scientists identified that we transmit more viruses to the animal than animals transfer to us. I just want to brief what is a virus, virus structure and composition. You see, this is a typical structure of the virus. They have a spike protein, they have a nucleic acid, they have a membrane, they have a envelope. This is a genetic material of the virus. This is, for example, the SARS coronavirus. They have different kinds of proteins here, and they have a nucleic acid. How the virus replicates, I just want to explain. So you can see that how the virus replicates. So first of all, this is the virus. They bind to the receptor in our skin, in our body. There is a receptor. So the virus binds to the receptor, and they are inside. And then there is some process going on, like translation and so there is some process going on, and then finally this virus can come here, and they will be released from the infected cells. So after one infection, the millions of viruses will come from here, from entry, and this is the active process. These are the targets. These are the different viral replication cycles, and there are different targets where we can develop the antiviral drugs. So these are the all targets it is reflecting. So now hepatitis C virus replication and immune evasion. This is a very important, because viruses are also becoming very smart. Like we humans become smart. We are using the artificial intelligence, and we are also using the smartphone. The same way the viruses are also very clever. They don't want to be recognized into the infected cells. They infect the human, they want to hide. They have a lot of skills, So this is an innate immune evasion. And these are, you see that this is a RIGI. RIGI is a virus scanning radar. This is a radar in our body. This is a protein, and this is always do the scanning of the virus sensor. So now I have one question. We also have a DNA, RNA, protein in our body. The same way, viruses are also composed with protein, DNA, RNA. So why the virus can infect our DNA, RNA cannot infect? So this is called the self versus non-self. So the any foreign particle that comes in our body that causes the infection, but we do have the same thing, DNA, RNA, protein, but we are not sick because it is a recognition. So RIGA is the responsible protein that works like a radar. So I just want to say about the antiviral targets. So some antivirals are targeting the cellular factors and some are targeting the viral factors. So these are different inhibitors. Proteases, RNA dependent, RNA polymerase. So these kind of different antiviral targets work. I would like to speak on repurposing drug. For example, we are taking paracetamol for the fever, anti-fever, but you never know that what paracetamol can also cure, or they can stop different kind of a diseases. So that is a clinical research that most of the doctors, clinicians, they have to do the repurposing drug development. For example, they can do a lot of studies. If paracetamol can also cure cancer, you never know. Because paracetamol can work against the cancer. It can be highly possible. So that's why it is called the repurposing drug. Repurposing means there is another purpose. It was developed paracetamol as an anti-fever, but now this drug can also work against the cancer. So that's why this is a way. But once you find this drug candidate that they can work as a repurposing drug, of course, the process will be very easy. You don't need to go for a very tough and complicated regulatory process because the drug is already in the market. All the toxicity and formulations and everything is defined. Only you have to integrate very fast, and FDA approval is very, very easy. If some drug is already in the market and we want to indicate that this drug can cure another disease, so that is called a repurposing drug. And during the COVID-19, it was all the scientists were trying that one drug can cure, for example, dexamethasone. Dexamethasone is not anti-COVID drug, but scientists were trying to prove that dexamethasone or chlorhexidine, they can work for that. So this is called a repurposing drug. So during the COVID-19, this is a very, very strategic way that we can develop some kind of a drug that can stop some kind of infection. For example, the rapid PCR, So this is all the things that can immediately start helping. I would like to bring your attention towards the status of WHO priority pathogens vaccines. So there are a lot of vaccines in the clinical trials, like Ebola virus. We don't have the vaccine approved. So it is still in the clinical trials. Marg is a very large pharmaceutical company. They are developing. Also the Marburg virus. So the vaccine is still in the clinical trials. These are they are in the different phases, So it is not completed, it is not approved. So different, for example, the plague, CCHF, Zika, Rift Valley fever, chikungunya, hantavirus, Marburg viruses, So if this vaccine, if this vaccine is approved, then we could be able to control the hantavirus. But we did not. Hantavirus did not come to India. I think few cases were there, or even not, I don't know. But if the hantavirus came here in India, we were not able to protect any population because the vaccine is not developed. It is still in the phase 2 or 3. So this is all milestones blueprint pathogens is doing. Now I would like to stop, but I would like to give your pandemic preparedness. The WHO CP also 100 days mission, European Center for Disease Control and Prevention, even the United Nations. These are the control, agencies. They have to work for the pandemic preparedness. They have to make the policy. They have to release the funds. They have to deploy the scientists and experts. Thank you very much. And last but not the least, to bring in your attention few scientists. He is Charles M. Rice. He is at the Rockefeller University, and he is expert worldwide on hepatitis C virus. So all the hepatitis drug vaccines, what we are trying, this is the scientist, and he is located in New York, and he is one of the scientists. He expert in the HCV. So he's trying to develop a vaccine for hepatitis C, but until now we don't have any vaccine for hepatitis C. For hepatitis B we have, but for hepatitis we don't. So he's a scientist, he's working on it. You can see that he's also the hepatitis, and he's in Heidelberg in Germany. Ralph Bartelsbacher, he's a very reputed virologist in Germany. So I have also worked opportunity to collaborate with him. So he's also a HCV guy. So you see that he passed away, but he's a Nobel Prize of papillomavirus, human papillomavirus. He was a discoverer and he was a Nobel Prize winner in Germany. And here is COVID-19. You know, she is a discoverer for the vaccine for COVID-19. And recently, we also have the antiviral drugs. He is Stephen Born. He is also in the virology department in Heidelberg in Germany. So there are notable virologists in Germany. They are trying to develop the vaccines and antiviral drugs and diagnostics. The experts of diagnostics are here. So now I give the opportunity to work to Thomas, that he will explain about all the technology he is bringing to it. And this is the organization I have worked. For example, I have worked with the Rockefeller University in New York. This is the CDC, Center for Disease Control and Prevention. I have also collaboration with the Nationwide Children's Hospital. Here is the Ludwig Cancer Center in Belgium and Cambridge. This is the Diagnomed Medical Devices. This is my company. This is the institute where I did my PhD. This is the institute where I did my postdoc. This is the institute in Switzerland. I did my first postdoc. And he is one of the guys. His name is Gunther Hartmann. He's one of the physical scientists. He's a discoverer. I mean, top oncologist, top immunologist, top antiviral drug. He is located in Bonn. Thank you very much.
Presentation. Ladies and gentlemen, scientific innovation achieves its true impact when visionary ideas are transformed into practical solutions that benefit healthcare system across the world. Our next speaker has been the forefront of precisely such innovation, contributing significantly to the advancement of molecular diagnostics through leadership, entrepreneurship, and technological excellence. It is my great pleasure to invite Mr. Thomas Green, Managing Director and Co-founder of Anchor Diagnostics, Germany, to deliver the keynote address.
Thanks a lot for the kind invitation. It's a pleasure for us to be here and to present our company in the next couple of minutes. For us, my colleague and me, a very intense week comes to an end. Unfortunately, we already have to leave India this evening. We had a lot of very interesting and intense discussions, and at all places and with all people we spoke, we always got the feeling that we are very warm welcome, and it was really a pleasure to be in your country, and thanks a lot for all of you. I would like to start with a short introduction about the company. Afterwards, my colleague will give you some more insight into our technology. And already the headline tells you a lot of our idea, what we would like to achieve with Anchor Diagnostics. As I said, I will shortly give you an introduction, that TOBIAS will tell you something about what we offer for molecular diagnostics, for the whole workflow, but also especially for the assays, the products that we offer, and hopefully could also, in collaboration with some laboratories in your country, could offer to your country. And then we would like to close with some key benefits that we believe our products could deliver. What is our mission? I'm meanwhile in the business of infectious disease diagnosis since more than 26 years. I started my work at the Diagnomed Institute for Tropical Medicine in Hamburg. This is one of the most important institutes for tropical medicine in Germany. And at this time I founded with some colleagues my first company. This was also focused already on molecular diagnostics of infectious diseases. We sold this company in 2005 to QIAGEN. QIAGEN is still the largest biotechnology company in Germany. Maybe some of you also know about the company because you see also a lot of laboratories in India, some of their products. At QIAGEN, I became the responsible vice president for the real-time PCR-based infectious disease unit and also the managing director of the facility in Hamburg. At the beginning of 2014, then I decided to leave the company, and I always thought about, during the time at QIAGEN, but also afterwards, about possibilities to further improve molecular diagnostics. As everybody of you at least knows, during or since the pandemic, PCR is a very powerful tool, but it's sometimes also long-lasting, and you need some expert knowledge to work with this. What we would like to offer is a reduction of speed, time to diagnose important infectious disease targets. With this, we would like to improve the management of patients, to speed up the whole process, and offer to the laboratories that use our products more flexibility and also improvement of the organization. That's what we have in our mind. As I already said, speed and flexibility is the key point that we would like to offer with our solutions. We think always about innovation to further accelerate this approach. And the customer is always in the center of our mind. We would like to discuss with our customer what they need, how could they improve, how could they also accelerate their idea to provide solutions to their customers, the patients, all of us. That is what is important for us. We founded the company in 2015 myself together with a colleague of mine, Dr. Rainer Zoller, and we started also immediately to develop our own quality management system, because quality is the key for infectious disease. We are selling our products to laboratories that provide results to patients. And it is always important that the patient, so all of us, could rely on the outcome of the study. So therefore, this was always important. And then in 2018, we launched our first product. As you could see, from 2015 to 2018, it's a long-lasting time, but we invest this time to develop the basis of all of our products, the chemistry, what the technology that makes our assays work. As I said, we try to speed it up. In 2020, or with pandemic, the Diagnostics got some boost. So we have to focus on COVID-19 detection. At this time, we did nothing else than COVID. So there was no possibility to develop other products. On the other hand side, we learned a lot regarding the production and the acceleration of speed. Between 2020 and 2024, we moved into a large campus. We developed new products. We also started to develop some so-called multiplex assays, so assays that could provide with one reaction more results. So additional advantage. In 2025 then, meanwhile we expanded our team up to 35 people. We tried to further broaden our portfolio with some PCR assays. And at the beginning of this year, we also launched an important additional step beside the PCR assays. This is the speed pure and the INTAKHAB. TOBIAS will guide you through. It's the initial first step. Before you use a PCR, you have to purify the nucleic acid that are used for PCR. At the end, as I said, the idea is to speed up the whole process as much as possible and make it as much as easy to the customer, so the laboratories, to use it. With this in mind,
Ladies and gentlemen, having explored the scientific vision and technological foundation of ultra-fast PCR, we now move to the next session, which focuses on its practical application in clinical laboratories and patient care. Ladies and gentlemen, having explored the successful adoption of any new diagnostic technology ultimately depends on its ability to support clinicians, strengthen laboratory workflows, and improve patient outcomes. Our next speaker brings extensive experience in precisely in this area. It is my privilege to invite Dr. TOBIAS RUCKES, Head of Sales and Marketing at Diagnostics, Germany. A molecular virologist by training, Dr. RUCKES brings over 2 decades of experience in molecular diagnostics, scientific communication, and laboratory solutions. Throughout his professional journey, he has worked closely with healthcare institutions and life science organizations in advancing innovative diagnostic technologies across diverse clinical settings. Dr. Tom.
Thank you very, very much for this kind introduction. Before I start, I'd like to first of all thank Dr. Alam and his teams, both in Delhi and Hyderabad, for this kind hospitality. It has been a real experience for us to spend this entire week from Sunday to Sunday here in this country. I know it's not enough. Everybody tells us this is a major market, this is a major country, it's very vast, it's very large. You need to spend at least 2 weeks to go to Mumbai and other places. I promise we'll come back at some point and then also see other places, but it was a truly impressive experience already now. We've had the privilege to talk to a number of people in prestigious organizations and institutions, and we're impressed by the experience, by the great questions that we were approached with, also some challenges that we were approached with. So we really appreciated that, and I think it's worth maybe repeating that at some point in time. Okay, you already told the audience that by education was a virologist, and I'm very proud to have joined this company approximately 2 years ago, and helping the organization Diagnostics to expand its footprint all across the globe. And this is the reason also, among others, why we're here to talk to Dr. Alam and his team at Diagnomed and Diagnostics in Hyderabad to see how this can be working, how we can jointly, bring our technology to market. We do the same in other parts of the world. We're currently working in Latin America through Colombia to establish a presence there in Latin America, and in Europe, of course. So we're trying now to test the waters in many different countries, and very happy to see the positive feedback that we receive on what we do here in this beautiful country of India. Right, now let me actually guide you through a few more technicalities of the Diagnostics philosophy that Thomas pointed out already. Is all about reducing speed, or actually increasing speed, and reducing complexity. That is the philosophy of everything that we do. And this has resulted in the development of a workflow for molecular diagnostics that can yield results in less than 60 minutes. And let me just outline this in some more detail to you. Let's cut a longer story, and actually molecular diagnostics is usually a longer story, rather short and simple. Now, there's a couple of details and technical details. I'm not sure if all of you will benefit from that, but some of you may. So I'll try to keep it reasonably detailed, but not too detailed, okay? So we start off with any patient sample in molecular diagnostics. Well, the first thing that you have to do before you do PCR, that Dr. Alam has referred to, you need to extract nucleic acids, DNA or RNA, from your patient sample, because otherwise PCR does not work. Think of SARS coronavirus. If I would take a nasal swab or throat swab and put it directly into PCR, I tell you what happens? Nothing. Nothing will happen. The PCR actually cannot work because it needs to have access to the genetic information of the virus, of the pathogen. And in order to get that access, you need to isolate the DNA or RNA in question. That's the first step. It's absolutely vital to molecular diagnostics. In the second step, you need to put together what you have isolated from your sample with the PCR reagents, and that's what we call PCR setup. And finally, with the help of an instrument, you detect then your actual pathogenic genetic information, DNA or RNA, by PCR, by polymerase chain reaction. And this process, still today, even though PCR has been invented, decades ago, still today takes an absolute minimum of 2.5 hours. That's the most ideal situation. It's actually regular, more in the range of 300 minutes, okay? Or sometimes even longer, depending on the on the laboratory's workflow, depending on the instrumentation used, etc. But we're quite proud to have brought down this entire workflow to under 60 minutes. And not only to reduce the speed, but at the same time still allow for full flexibility in the laboratory. And that's, I think, the right combination. Reducing speed, I don't know why I keep saying reducing speed. Increasing speed, reducing complexity, maintaining flexibility. The workflow looks like this. This is now a little bit of a product slide. The workflow starts with, as I said, any patient sample, and it then is subject to a nucleic acid extraction, DNA, RNA extraction, as I outlined earlier, with the help of this little machine here that I'm going to introduce you to in a moment, and these reagents that are proprietary development from our company. This combination here of nucleic acid extraction takes you 15 minutes. That's really fast. There are others that are also fast, but not as fast, and certainly not at this quality level. After the nucleic acid extraction, then the PCR can take place with the help of our PCR reagents called anchor PCR kits, and they will take you only 30 minutes. And just Thomas outlined, this is where we started. We started with anchor PCR kits. We brought down the result time, turnaround time to under 30 minutes, and now we're able to bring the entire workflow time down to, as you see up here with our clock, to less than 60 minutes. Now, how does that work? The anchor beads works always in combination with one of these different formats of what we call the SpeedPure technology. SpeedPure is our brand name, and basically includes a chemistry that we have very sophisticatedly developed to bring down this isolation time to approximately 15 minutes. And then, as I said, the PCR kits thereafter. Let's go into a bit more detail. First of all, you need to understand this, the basic principle, the scientific principle behind this nucleic acid isolation, is magnetic bead technology. So what we work with is this magnetic particle technology. It's not invented by us, that's long in the market. We've just made use of it and maybe optimized it a little bit. So you start off with a lysis buffer that we provide with our reagents, and what you add is your patient sample, for instance, whole blood, or serum, or cerebrospinal fluid, or swabs, okay? And thereafter, you add another reagent. It's an enzyme called proteinase K that we also supply. It's actually added to make this lysis of your sample even more efficient. So this is what happens next. The lysis takes place, the cells will be opened up, the genetic information, the DNA and RNA is released from the cells, right? It's then available in this liquid. And now here comes the machine, right? The machine, it basically is nothing else but a machine that transfers these magnetic beads from one place to another. Magnetic beads, I mentioned earlier, are essential to our technology. What happens is that the machine actually has these magnetic rods, and together with the magnetic rod cover, goes inside this solution here and absorbs the nucleic acids. They bind to the magnetic beads, and then the magnetic beads, with the help of this magnetic rod, are being transferred to other wash buffers, to other solutions. They are washed. Why is this washed? Because there's no impurities in there, inhibiting substances. You need to remove them all before you eventually elute your DNA or RNA question. That's the startup process. What we have done is this bit here down there. We have made the lysis cross step particularly efficient. And we did so because we combined enzymatic lysis with thermal, mechanical, and chemical lysis. So the way we've optimized that in our reagents, I think, is unprecedented, and that allows us to do it very efficiently and, by the way, with one universal protocol. You don't need different protocols for different specimens, you don't need different protocols for DNA or RNA, it's all the same. And that goes along with our philosophy of reducing complexity and making things straightforward and particularly simple. This is the machine. We have a look inside the machine. The machine allows you to do up to 32 samples in one run. And it doesn't matter if you run one sample, or 5, or 10, or 17, or 32, it almost takes you 15 minutes. The result, the instrument is like this big, right? It's like a cube. It fits into every corner of a lab. It's very small, and it is a very standardized workflow, as I said, universal for any type of sample. And actually, you might think, well, 1 to 32 samples, not that many. I have a higher throughput, or I have different needs, sometimes more, sometimes less. The good news is that this is very adaptable. So you can run 15, 16 samples, say, as an example, right? It takes you 15 minutes. Or you could run 32 samples with a low to medium throughput. Or if you have like 48 samples, as just an example, it might take you 30 minutes. So it's not that much longer, right? And if you have even 96 samples in one go, well, it would take you 45 minutes. You just run the instrument 3 times in a row. So it's very simple and efficient. And again, one universal protocol. The instrument is simply operated through a touchscreen. Up here on the right-hand side, there is an LED lamp, so even if the lid is closed, you can see what happens inside the machine. There is a UV lamp built into the machine that you can activate to, prevent any contamination. There's even HEPA filters on the top. And as I said, it's very flexible, universal protocol. We talked about the size already. Now, I have brought along a little video, which I'd like to show to you. Just give me a second. I hope that's...
Life pulses fast, it accelerates, busy and vivid, hardly any time to pause. The same is true inside today's laboratories. The pace is just as intense. More samples, tight timelines, rising cost and increasing complexity, but limited resources. Time is of the essence. Every minute counts. What if molecular workflow routine was simpler, seamless, and ultra-fast? We are introducing ANCHOR-Bead 32, a nucleic acid purification system that turns lab stress into seamless efficiency. Paired with speed-pure reagents, the ANCHOR-Bead 32 platform allows DNA and RNA extraction of 1 to 32 samples in just 15 minutes from a wide variety of samples. In combination with high-speed ANCHOR PCR kits, this powerful workflow delivers full results in under 60 minutes, precision without the pressure. Empower your team to handle more, faster, smarter. Reclaim time and flexibility in every run. Because the future of molecular workflows shouldn't take hours, it should take under 60 minutes.
So actually, it's a really neat and importantly also very robust machine that we have in operation now for many years. Actually, 3 of them in our departments, and we haven't had a single failure. So they are very low maintenance, very simple to use, and especially when used in combination with the speed pure reagent. I mentioned earlier the speed pure reagents is our own proprietary development, and we're proud actually that by using those speed pure technologies, we can bring down this nucleic acid extraction time to about 15 minutes. It's available in 3 different formats: SpeedPure Go kit, which is the cartridges that you see here on the left; SpeedPure Multiplate, which is in the middle for medium throughput. If you have batch samples, number of 16 can be done with 1 plate. Or if you would like to have flexibility, you take the SpeedPure Flex kit, which is bottled reagents, and then you can self-dispense into deep well plates that we also provide. So we basically have covered any sort of throughput need in the lab. If you have 1 or few samples, you take the cartridge. If you have batch samples, you take the plate. Or full flexibility, also possible in combination with other instrumentation from other vendors. Not that we would advocate for the want to promote that, but it's possible. The SpeedPure Flex version on the right hand side. What do you find in such a cartridge in our chemistry? In a Go kit, just to take that example, you find 48 of these cartridges, and they contain lysis buffer, wash buffer 1, wash buffer 2. These magnetic beads that I mentioned earlier, which are absolutely essential to the entire procedure. Then an empty cavity, which we do not need, and an elution buffer. All of this is pre-filled and sealed. So the only thing that you basically have to do is peel off the seal, add your sample, and your proteinase K, as I outlined earlier, and basically put it inside the machine. So it's very straightforward. The proteinase K that I mentioned is also delivered as a separate item. And then you find these magnetic rod covers. These are little sleeves that you need to put inside the machine to protect your magnetic rods from contamination. So they're also added, and every single kit features a quick guide that tells you basically at a glance what to do. So make it very straightforward to use. And of course, the same setup or similar setup applies then to the other kit formats. We don't need to go through this right now, okay? So it's all basically already pre-configured. Okay, now how does that work? What do you need to do for a sample preparation?